Thank you so much for inviting me to be here. My path to being where I am is a little bit weird. I was an undergraduate here at University of Georgia, and I studied lots of different things. I didn't actually declare a major until almost the end of the fourth year of my undergraduate degree, when I discovered that all of the cool stuff that I'd been doing actually led to an ecology degree, which was cool. And so I got a degree. I worked in Pick's lab, which taught me that pretty much anybody can be a successful scientist. Just kidding. Pick, are you here? I told him I was going to rib on him just in the beginning to see if he would come, right? He's probably not here. Right. So I went to Oxford to do a PhD, and I was really interested at the time in evolution of sex, and I wanted to work with Bill Hamilton. But Bill got malaria and died of complications of malaria a few weeks before I ended up in the program. And I was walking through the hallway at Oxford, wondering what I was going to do with the next 4 years of my life, when I met this irreverent New Zealander, Alan Cooper, who said, "You can join my lab. I just got here. I don't have any students. And if you join my lab, you can go to Siberia." And that is how I transformed from a community ecologist who studied tropical insects to a geneticist who works on extinct Arctic megafauna. But I wouldn't have it any other way, right? So when I was thinking about what I would talk about tonight, I thought that what I would do is I would spend this evening, after a nice long day of reminiscing and talking about science and going back to old times, to champion us and what we do. Because let's face it, we are not exactly basking in the love of the powers that be right now. And I realize that I am speaking to the audience a bit, but we all every now and then need a pat on the back, right? And after all, what I do seems so completely irrelevant to the problems that we're doing today. I thought if I could show how the stuff even that we've discovered by extracting DNA from the bones of mammoths was relevant to solving the problems that we're facing, that we could all start to feel a little bit more confident in that what we are doing, you are doing, which is clearly more relevant than what I am doing, is really awesome, and that we could go back to fighting the good fight. But as I've talked to people about this over the last few days, I've discovered that people are disappointed with this idea. And in fact, most people were expecting that I was going to give a behind-the-scenes update about how close we are to bringing extinct species back to life. Which is totally fair, because I did call my book "How to Clone a Mammoth," I don't want to disappoint you after dinner, right? So I'm going to do both, right? I'm going to start with an update on de-extinction, which is the terrible word that people have chosen to use to mean bringing extinct species back to life. It really is terrible. How do you conjugate de-extinction? Seriously. You say that I have de-extincted something? Please don't say that. Right? So I'm going to start with that, and then we'll get on to some kind of light after-dinner science. I have one data slide, so that's light science, right? One data slide. So first, de-extinction. This is actually weird. It's a really good time for an update on de-extinction, actually, because just yesterday, it was still not possible to bring extinct species. And so that we are totally clear about this, the list of things that I have been asked, if are coming back to life, that it is still not possible to bring back to life includes mammoths, passenger pigeons, Neanderthals, Hitler, the dodo, Harrington-Hippus francisci, which is an extinct species of horse that you've probably never heard of because we only named it about a month ago. It's pretty cool. Died out about 12,000 years ago, lived at the same time as regular horses that we like. Saber-toothed cats, any kind of dinosaur, just no, right? The Tasmanian tiger, the great auk, the gastric brooding frog, which that would be my first choice, because how awesome is a frog that swallows its offspring and then barfs up fully formed juveniles? My choice, seriously. the Carolina parakeet, and Max, who was a Keith hound that was in my house when I was very young, who apparently when I was a toddler would let me stick my pinky up his nose and not bite. That's right. All of these things are dead. They are gone. They will not be able to be brought back. They are going to be gone forever. I'm sorry. But seriously, this is actually, I'm gonna hover on this for a minute and just try to hover in the spotlight. I keep moving, but I've been told that I should stand either over here where you can't see me, or over here where you can probably see me. Anyway, this is something that concerns me a lot because I get a lot of calls from the popular press. Every time George Church says something, I get a call from the popular press that says, oh, we're gonna have mammoths in 2 years. Well, elephants have a 2-year gestation, so probably not, unless there is a gestating mammoth somewhere, which there isn't. So this concerns me because I can't help but think that the more we talk about de-extinction as if it really is something that can happen, the more flippant people are going to be about extinction happening. And I think we need to be very clear that once a species is extinct, we can never bring back an identical copy of that species. From a technical perspective, this is because there are teams out there around the world that are working on this, supposedly bringing back mammoths or Tasmanian tigers or passenger pigeons. But none of these groups have living cells from any of these species. And that's because there is no living tissue from any of these species. And if one is going to bring a species back to life, the main technology to do this is cloning, a la Dolly the sheep, right? But you can only do that if you have a living cell. Now there's lots of genetic material, my career is based on this, from these things that are extinct, but that DNA that we recover from these remains is broken. It's physically damaged, it's chemically degraded. One cannot clone something from a broken, physically damaged, chemically degraded cell. Cloning is not possible. Extinct species are going to remain extinct. Of course, there is this new super cool technology of genetic engineering, genome editing, using CRISPR-Cas9, this beautiful molecule right here, where the idea here is we can extract DNA from a whole bunch of mammoth cells, we can extract DNA from a whole bunch of elephants, we can line up these genomes in a computer, we can figure out exactly where these species differ from each other, and then we can use a cut and paste kind of approach to swap out bits of elephant DNA for mammoth DNA. And we can do that. We can do that. We can't do it for all 1.5 million differences right now. That's a technological hurdle that we can fix. But in the end, the thing that you end up with is an elephant cell that's been slightly modified to contain some mammoth genes. Maybe we can take whatever that thing is, this hybrid mammoth elephant, and we can use it as a proxy for the thing that's gone. But it is not a mammoth. In case you were just curious about that. It's not a mammoth. Extinction is forever. And the kind of field that I'm in, ancient DNA, is cool. We do a lot of cool stuff, but we cannot reverse extinction. And I think that this is an important thing that we should all remember. There are lots of other things we could talk about. Technical, ethical, ecological challenges associated with de-extinction. I'm not going to talk about that, though. I am going to talk about some of the stuff we can do. And while we can't bring extinct species back to life, we can try to understand why species became extinct in the first place. And this really is the motivation for my research. That if we can understand why species change, why communities change, why ecosystems evolve, what happens when these large-scale disturbances like massive climate change, global warming, glaciation, when these things happen, how do they affect communities and ecosystems? Maybe we can learn from the past things that we can apply to the decisions that we're making today to protect species and communities and ecosystems that are alive today, but in danger of becoming extinct. So I work in a part of the world called Beringia. Beringia spans from Alaska, the kind of mountain range in Canada's Yukon, across that white area, that's the Bering Sea, into the part of the world that, in kinder, gentler political times, Sarah Palin could see from her backyard. Do you remember that? Do you notice that it's white there? That's because the sea level stays a lot more shallow there. So during ice ages, when much of the water on the planet was taken up into making big glaciers that sat on top of the continents, that was exposed. And it was a bridge, a land bridge, a very productive land bridge that was able to support huge numbers of plants and animals. Today it looks like this. I'm actually in that helicopter taking the picture, and I'll show you a picture of that helicopter in a minute, because it's awesome. But in the Ice Ages, it looked more like this, where you had a rich community of carnivores, like these saber-toothed cats, and there were other cool giant bears that stood 16 feet tall, called Arctic, short-faced bears. I don't know why they're called short-faced bears. You'd think if you had a 16-foot tall bear, the thing that you came up with for its common name wouldn't have to do with how short its nose was. And then you had all these lovely things that we associate with the Ice Ages. Mammoths and mastodons and woolly rhinos and camels and lots of different species, of course. It was awesome. Much more awesome than it is today. We saw that picture a minute ago. So we go out there. There's the helicopter. That's good. You see that some of the windows don't actually have plastic or glass in them? That's good. That's good because this helicopter, I got this helicopter in Khatanga, which is the name that is written on top there, which is this horrible town in sort of north-central Siberia. And it took off the third try. We loaded it up and unloaded it. And then several days later did it again. And then several days later did it again. And finally it took off, right? And as we were all nestled in there with all of our crap for being out in the field for months, sitting on top of these massive gas tanks that take up most of the inside, when it finally took off, the French and Russian expedition team leaders decided they would celebrate the fact that we had taken off by smoking. Right? So it was really great that there was actually some room for air to come in and out, right? And we stay in 5-star accommodation. I worked in the tropics before when I was here, and I thought the bugs were bad there. Holy shit, the Arctic. Those are mosquitoes. I backed up from my tent and unfocused the lens so you could get a feel for the depth and fields of mosquitoes that you deal with when you're up in the Arctic. It's awesome. And we wander around looking for bones. So this is actually near Dawson City in the Yukon, in the Klondike. And the Klondike is the gold fields that are up there. And the gold miners are washing away that frozen dirt to try to get to the gold-bearing gravels that are underneath. And as they're doing this, thousands, tens of thousands of bones just fall out of the permafrost, and they're frozen. And these are bones that date back to the last 1,000,000 years. The oldest bones we found actually are about 700,000 years old. We know that because they're associated with a volcanic ash layer that's been dated to that age. And so we wander around. These are people from my lab sort of wandering around here, picking up bones as they come out. And we take a chunk of, in a typical day out there, we'll find maybe 20 or 30 bags like this. So there really are just, I teach a class where I take undergraduate students up there and I say, don't worry about it, you'll find a mammoth bone. They're like, no, we're not gonna find a mammoth bone. And they're out there for 5 minutes and they're like, oh! Mostly it's bison, actually. Most of these things are bison bones. Bison were the cockroaches of the Pleistocene. There's bison, and there's horses, and there's caribou, and every now and then we find carnivores like bears and lions, and it's very cool. And we take a chunk out of these bones in the field, and we take them back into our lab. We have these super fancy clean lab that is basically, it's just like one of these labs that you have to protect yourself from getting horrible diseases, except we turn the air pressure in the opposite direction because we want to protect the bones from being contaminated by us. So we dress in these funny suits, much to the amusement of anyone who wants to put us on TV, and we cut out big chunks of bone here. And from that we extract DNA. So, anybody who's been following the world, like you guys have, has seen Michael Mann's hockey stick graph here. This is a plot over the last 1,000 years of the average global temperatures. This is not my data slide. This is not my data. No, this is not my data slide. Average global temperature over time. See, it's been, if you squint, maybe declining a bit, but then skyrockets up here after about 100 years ago. We've also seen projections that go off the other side where it goes up into the stratosphere and keeps us up at night. And this we're very concerned about. But this is not the first time in Earth's history that the Earth has suddenly warmed. So if we take the same sort of measure, here again a proxy for average global temperatures, but going back 100,000 years instead of 1,000 years, what we see is that Earth's temperature was crazy for a while, and relatively recently, I'm going to focus your attention here, it increased really quickly. This is the transition between the cold ice age, the Pleistocene, and the warm interglacial period, the Holocene that we're in today. We have a site in the Klondike where we were showing you, I showed you spraying down those bones, where you can see that transition in the dirt. You can see that it goes from this dry, there's nothing there, definitely Pleistocene stuff, all the trees and shrubs start coming in. And that transition, this transition, which is on the order of about 8 degrees Celsius, we believe, based on these radiocarbon dates, happened within around 30 to 40 years. That is a very rapid global warming event. And so what I do in my lab, what people in my field do, is we go back over this time period and we collect bones that date to this time period and we ask these questions. How did species and communities and ecosystems respond to this past period of global warming? With the hope that we can learn things that we apply to today. So I'm going to tell you 2 quick stories about things that we have learned so far and how it's potentially relevant to today. And hopefully they won't be boring and they won't be too long, so everybody can get back to having a nice conversation and not listening to me. First, mammoths, because people like mammoths. As soon as you find out that you can't bring dinosaurs back to life, people want mammoths. I don't know why, but that's totally true. If it's not dinosaurs, fine, mammoths. So mammoths actually went extinct relatively recently. Probably more recently than most people think, most people believe. On the mainland in Asia and in North America, based on DNA that we get directly from dirt, we think that mammoths survived until around 8,000 years ago, but there were 2 populations on 2 islands that survived much more recently. On Wrangel Island up here, this is the last surviving mammoth populations. Mammoths lived until around 3,000 years ago. That's pretty recent. And on this island here, St. Paul, which is part of Alaska, they survived until around 5,000 or 6,000 years ago. And so one of the projects that I've been involved with for the last couple of years is to try to figure out, focusing on St. Paul, because we know a little bit more about Wrangel, focusing on St. Paul, exactly when and hopefully why mammoths went extinct on this island. So we were asking this particular question. St. Paul is a funny place. The nice thing about St. Paul is that we can rule out that people made mammoths go extinct, because people did not arrive on St. Paul until the 1700s, when a bunch of Russians kidnapped a bunch of people from the Aleutian Islands and forced them to move to St. Paul and club seals. True. They also brought caribou. Right? We always think about the reindeer on St. Paul as this amazing example of carrying capacity, where the reindeer could totally exceed their carrying capacity and eat everything and then nearly go extinct. Those reindeer were introduced by these same Russians that brought the people onto St. Paul. It has 1 town, 1 church, 1 store, 1 bar. It had a population of about 483. While we were there one summer, there was a baby born, there was a party around the sign, and they changed the number. Like, this is a small town. St. Paul became isolated from the Alaskan mainland. This is presumably when the mammoths became isolated from the mainland population around 13,500 years ago, very early in the transition between the Pleistocene and the Holocene. It shrank over time as the sea level rose, and about 6,500 years ago, it got to its present land area. We know mammoths survived for some time after that, and then people were introduced in 1787. So we went out, and we went to this place on the island called Lake Hill, which is right there. It is a volcanic caldera. It's the only source of fresh water on the island. And during the winter, when everything was frozen, we shoved some holes down into this lake, and we collected dirt samples that went back through time. Here we are doing this crazy thing. It was terrible. Why do people live there? I used to work in the tropics. What did I do? Why am I doing this now? Anyway, yeah, it was cold. So we went out there and took this lovely core, and then we took it back, and we took samples down the length of these cores after we figured out how old they were. And from these particular samples, we processed and asked a whole bunch of things. We collected cladocerans and diatoms to tell us about how salty the water was, how turbid the water was, etc. We looked at sedimentary DNA. That was the part that I did, where we actually just extracted DNA directly from dirt and asked whether we could see mammoths or any other different species there. We looked at these fungi, Sporomyella, that only live on the poop of large mammals to ask whether that corresponded with what we see with DNA. DNA, and we did some isotopic analysis and some analysis of the magnetism of the soil, so we could try to really reconstruct what was going on in the climate of St. Paul Island over this time. And we actually solved it. This is my data slide. Only one. We solved this problem. We found out, and I'm just going to go through it quickly here. This is vegetation. The vegetation over the course of time here, from 11,500 years ago to the present day, does not change. There's no affected vegetation. Mammoths did not die because they ran out of stuff to eat. What does happen, though, is the lake becomes increasingly shallow and increasingly turbid and increasingly salty. Mammoths went extinct on St. Paul Island because they ran out of fresh water. And what we see here is that this is the DNA. These are the radiocarbon dates from bones on the island. We had a party, invited everybody on the island who lives there to our party, asked, bring us any mammoth bone or teeth you've ever found, and we will give you a radiocarbon date for it. They brought us tons of things. This is a great way to do science, just give people food. and we dated a whole bunch of bones, and we found out that the DNA here, we have yes mammoth DNA, and then suddenly no. It matches with the Sporomyella. This is the fungus that grows only on the poops of large mammals. It exists, it goes away at the same time as the DNA. It comes back when the caribou come back. It's pretty cool, right? So we've actually figured out why mammoths went extinct on St. Paul. They ran out of fresh water. The other thing that we learned about mammoths at this time, which is similar to what we've learned from Wrangel Island, where there are many more well-preserved bones, and now we have genomes, is that these animals were in bad shape. They were losing gene function. They were highly inbred. They were basically suffering from having been part of this tiny little isolated population for many thousands of years. And we see this in both places. Most of the genomic work has come from Wrangel Island, but these analyses have shown that by the end, these animals it was surprising that they could get up and move around and breathe. They were so broken genetically. And if we step back from that and think about what's happening in the other animals that we're studying, we actually see the same pattern across the mainland. What happens as we transition from the Pleistocene, where you have lots of habitat for everything everywhere, into the Holocene, where we start having these isolated patches of habitat, is that you see that within each of these isolated patches of habitat, you have populations that are becoming increasingly inbred, increasingly broken. And if they could move from one patch to another, we know it takes one generation of outbreeding to relieve the effects of generations of inbreeding. But they couldn't do that, particularly after humans turned up on the scene and knew how to block the movement between these isolated patches of refugial habitat. But we see this exact same pattern as extinction approaches for woolly rhinos, for horses, for bison, for lions, for mammoth and mastodon, for camels. This exact same pattern. If you sample broadly and you say, is the population okay? They look fine. I take one sample from here, and one sample from here, and one sample from here, they all look fine. If this individual could move over here, they would be fine, but he can't. And so they're not. So how is this applicable today? This thing that was happening in the past is very much what we see happening today. Human population growth, changes in land use, expansion, the building of cities and roads and dams and blocking rivers and things like this. We are blocking off populations from being able to move back and forth between each other and rescue the individuals from the effects of inbreeding. I have a project in my lab right now where we're working with California mountain lion populations. And we're looking at the genetics of mountain lions in Santa Cruz Mountains, Santa Monica Mountains, and other places across California. And if you look at each of these populations individually, you see incredible amounts of inbreeding. And we have a project with Chris Wilmers, who runs the Santa Cruz Mountain Lions project, who puts radio collars on these guys, and we trace what's happening. You have a good summer, like we did this year. You have a lot of mountain lion babies, a lot of mountain lion baby boys that are doing just fine, but they have to leave. They have to go somewhere. They have to find and establish their own range. And this is the kind of thing that happens. We have a mountain lion, a boy who's born here, who moves across here. This is San Francisco Bay. Moves across the Santa Cruz Mountains here. This is Silicon Valley. He's running around trying to figure what's going on. This is somebody's camera from the front of their house. Right? This is a mountain lion standing on a car in Mountain View, right? This is not normal, but it is normal. This animal was actually killed trying to cross the highway about 3 weeks after this picture was taken. And this is what happens to these animals. Is there a solution to this? There is. There are several solutions. We can build corridors. We can buy land that makes connecting corridors. We can even do translocations. We can move individuals from one population to another after we do a genetic scan and figure out how one individual is different from another. We know this works because it saved the Florida panthers from going extinct. When people moved in the 80s, Texas panthers into Florida. One generation of outbreeding and they lost the cryptorchid, they lost the crooked tail. They were able to survive. And we know that it works. This stuff is risky though. We gotta move individuals, we gotta think about changing the way we behave, and this could be a very long discussion that we could have here about relative risks versus rewards, and I'm not gonna get into that. What I am gonna do is show you this picture of all of these adorable baby endangered animals. And ask you to consider whether it is riskier to move animals around or to allow these animals to go extinct. All right, one more quick story. So passenger pigeons. This is the project that I've been working on for a very, very long time. Actually, I started working on this project when I was working on my PhD in 1999, 2000. One of the first projects I was involved with. Passenger pigeons are fascinating, right? This is a passenger pigeon. This is Martha. Martha was the very last passenger pigeon to be alive. She was in the Cincinnati Zoo until she died. She looks pretty sad here. That's not because she's the last one to be alive, that's because she's dead. She's been dead in this picture for a very long time. She died in 1914. This picture was taken in 2005. Right? But passenger pigeons, and Martha among them, were mostly famous because there was an enormous population of these birds. Enormous. And this is what they're famous for. In fact, the very first census of passenger pigeons was taken by French explorers in the Maritimes who were exploring, as explorers are wont to do, and they were looking at the sky, and they actually counted the number of passenger pigeons that were there, and they wrote down that there were an infinite number of them. These birds would flock. They actually did do regular census later, and they found that these birds probably were somewhere between 1 and 3 billion individuals. They flocked in a single flock. Sometimes it split. They would move from massing forest stand to massing forest stand and just consume absolutely everything in the wake. They would also kill the forests because of they would nest 500 birds on a given tree, and they would have so much guano that they would destroy everything that was there. They were the avian equivalent of an EF4 tornado tearing through they were and they were like that for a very long time until we murdered them all. Right? And note the log scale here. They went from many, many individuals to none over the course of several decades. And the reason is because we were very good at killing them. We would shoot them just randomly out of the sky. We'd put up these big nests and we'd catch millions at a time. They were the poor man's chicken, right? But then we ate them all, right? And they went extinct. So I was interested in passenger pigeons because I thought, this is insane. How could there be a bird that has this many individuals? That's crazy. And I wanted to try to figure out when they became so abundant. Because if I could figure out when they became so abundant, maybe I could better understand why they became so abundant, because from an evolutionary perspective, this is a very weird and interesting thing. And so I started collecting specimens. We went to museums. In fact, tons of the specimens that I got were from the Royal Ontario Museum. I have about 200 birds in my collection. Topads, little tiny topads. They're from the Royal Ontario Museum. Archaeological collections, different museums, and we got little tiny bits of bone and extracted DNA and identified individuals that are really well preserved so we could try to learn about these things. And as with any project that you start and that it takes a long time to do, a lot of things happened along the way. I got no funding for a while, so we didn't do that, and then I tried to discover things, we published a little bit of it, and then, we got scooped. Somebody published a paper in PNAS that actually had the first genomes of passenger pigeons. One morning I woke up to that. That was pretty terrible. Yeah, these are the things that happen, right? Fortunately, they got it really wrong, so that was okay. Turned out to be okay. We didn't publish a response either, because that's a silly thing to do. Instead, we waited several years so people forgot about that paper, right? And then we published our paper like a month ago. But anyway, what we have done, it was published a month ago. It was on the cover of Science. to get scooped and then get the cover science. So what did we learn about passenger pigeons? Well, the one thing that we didn't learn, the one thing that we did not learn is when they became so abundant. And this is because of all the weird stuff we did learn about the passenger pigeon. Here's Martha again, this is a thing from National Geographic. But what we learned was that passenger pigeon populations were as large as they were when Europeans arrived, at least since before the last ice age. This is an incredibly adaptable bird that is just able to succeed in whatever environment is thrown at it. But this, of course, having a very large population for a very long time has important evolutionary significance, right? And I'm looking at the students in the crowd here, because my students are here. Why? What happens? What is the most powerful evolutionary force when you are a very large population? Natural selection, right? Not genetic drift, that's powerful when you're a small population. Natural selection is a powerful evolutionary force when you have a big population. These birds, over the course of their existence, have become, because natural selection is such a powerful force, became excruciatingly, and excruciating is the right word because of what happened to them, well adapted to living in large populations. And when humans came around, and in 40 years reduced them to dozens of individuals, that was the equivalent of taking a freshwater fish and throwing it into the ocean and saying, okay, there you go, right? So being an incredibly large population, a successful species for a very long time, actually made them unfit when suddenly they became a small population. And this does have relevance to the way that we think about conservation today. And there's two ways that it has relevance. The first is that we often think of very big populations of things as off the hook. They're fine. They're not going to go extinct. That's not necessarily true, depending on the history of that population. And the second way is that when we make decisions about how we're going to conserve things, we often come up with some arbitrary number about what the population size should be returned to. And often that number is 500. It's just a number. I don't know where that's come from, but that's often the number. But if you are a species that's used to living in extremely large populations, for example, maybe 500 individuals or disconnected populations, bringing us right back to the mammoths, is not the way that it's going to work. And so by showing that we have this extremely large successful population that just can't do it after a short time should give us a little bit of insight into how we need to start thinking more holistically about plans to conserve species. Maybe we need to think about history a little bit more. It actually is relevant, looking at the past, to come up with ways that we're going to deal with species today. Now, I've said too much now, and I am going to be quiet now so that we can all get back to happiness. But what I just wanted to say was that even when the work that we do seems like it might potentially not be relevant to life, yeah, it's still worth supporting. It's still worth thinking about funding and doing. You guys are all doing really cool and really interesting things, and maybe it's basic science. Maybe it's going out and collecting data and contributing to long-term research projects. Maybe you're doing environmental consulting and you're adding to a big data set of resources that we have, or writings that we're dealing with politicians, or we're thinking about the ways that we're talking to each other. This is all important and vital work. And don't ever let anybody tell you that the stuff you're doing isn't interesting or isn't relevant or isn't cool, because it is. You guys are awesome. And thank you for listening to me.